Hello everyone, my name is Ian, and today I will introduce this paper, Evidence of Dark Oxygen Production at the Abyssal Seafloor. And in the past, people think that in the deep sea there's no sunlight and dark, and there's nothing. However, in this paper, the researchers found a surprising discovery. They found oxygen in the deep sea, and they call it dark oxygen. And first, we should know why the oxygen is important in the ocean. Because oxygen can control respiration and marine ecosystems. And the oxygen also can be an indicator of ocean health. And this picture shows the main character of this paper. This is polymetallic nodules, and also called manganese nodules. And this just like a stone. And it's formed of concentric layers of iron and manganese hydroxides around the core, and they grow very slowly, about 1 centimeters, over millions of years. And the size of these nodules just like a potato. And then this picture shows the polymetallic on the deep sea floor. They cover about 70% of seafloor, and they are common in Clarion cryptosome, also called CCZ. CCZ is the study regions of this paper. And the researchers believe that polymetallic nodules may be a potential source of deep sea oxygen. And then is the study region. The CCZ is located in Pacific Ocean, and they are abundant of nodules. So this is the reason why the researchers choose this place to study. And then is my methods part. I summarize the methods into 3 parts. The first part is field measurements. And the researchers will do the experiments on the seafloor. And the second and third part is shipboard incubations and laboratory analysis. The researchers will collect nodules from the seafloor and take it to the shipboard and laboratory to do the incubations. And then is in situ experiments. In situ means the measurements were conducted directly on the seafloor, and the researchers will use the benthic chambers. This is a device that will be deployed to enclosed sediment areas, and they will monitor the oxygen concentrations over time. And they also use the method microprofiling. This technique can help researchers to understand how the oxygen moves between sediment and seawater. And then is ex situ co-incubation. This method is opposite of in situ experiments. So ex situ means they will do the incubation outside of the nature environments. They will first collect nodules from the seafloor and test it in laboratory conditions. And they also monitor oxygen concentrations over time. And they also use the opto sensor. This sensor can record dissolved oxygen. And the results will be validated by using Winker titrations. And the researchers also want to know the source of oxygen. So they use the 2 methods. The first one is microbiological sampling. They will analyze the microbial community to determine the microorganisms can produce oxygen or not. And the second one is electrochemical measurements. They will measure voltage on the surface of polymetallic nodules and to determine whether electrical energy can drive erection seawater. And both of these methods is to determine is oxygen produced by microbes or electrochemical reactions. And then is my results part. These figures shows different cruises, and that shows oxygen concentrations measured by calibrated oxygen apoptosis. And the x-axis represents incubation time. Y-axis represents oxygen concentrations. And different colored lines represent different treatment conditions. For example, the green line shows dead algal biomass, and the red line represents filtered seawater. And after the experiments, the researchers found an unexpected oxygen increase. We can see in the figures, all the line is increased. So we can say under different treatment conditions, the oxygen concentrations showed a net increase. And this provides direct evidence of DOP at the abyssal seafloor. And then its oxygen concentrations measured in different regions. These figures, x-axis represent incubation time, and the y-axis represent oxygen concentrations. And all the regions is in the CCZ. And we can see all the lines is increased. So we can say DOP is supported as a widespread phenomenon. And this figure shows total net oxygen production in ex situ sediment cores. And in these figures, x-axis represent treatment conditions, and the y-axis represent DOP. And we can see all the data in the figures is positive. So under various treatment conditions, oxygen productions is be seen. So DOP can be observed in ex situ incubations on the ship. And then the researchers try to rule out biological oxygen production. And how they do that, they added mercuric chloride to seafloor sediment cores. Mercuric chloride is this substance, and this is toxic, that can kill all living organisms. And watch these figures, we can see the data still is positive. So the result is DOP was still observed in this dead cores. And this proves that DOP is a geochemical process, not a biological. And these figures shows background corrected voltage potentials on nodule surfaces. In these figures, y-axis represent voltage, and the units is volts. And this is a box plot. All the nodules is under 21 degrees, except for the 6 and 7 nodules is under 5 degrees. And we can see the x represent means, and the line represent median, top and bottom of the box represent quartiles, which curves represent minima and maxima. And see the figures, we can find that the electrical potentials on all the nodules. So the researchers think that the electrochemical reactions may occur on these nodules. And this reaction may generate oxygen. And then it's my first discussion. In the previous page, we said that the electrochemical reaction can generate oxygen, and how they do that. The researchers think that is water splitting. However, they meet a challenge, because theoretical electrolysis of seawater requires 1.23 volts, but the measured voltage on the surface of the nodules only reach 0.95 volts. This is not enough. So the researchers assume the explanation, the catalyzed effect. They found polymetallic nodule contains transition metals, for example, manganese oxides. And these metals act as catalysts to lower the energy barrier and make the water can be split and produce oxygen. And it's my second discussion. In the past, deep sea ecosystem was thought to rely on the sinking organic matter. However, in this paper, DOP could provide an additional oxygen source for the organisms in the deep sea. And that may influence deep sea biochemical cycles. And the last is my conclusions. In this paper, oxygen production was observed at the deep sea seafloor. And the researchers also do many experiments to confirm the presence of net oxygen generation. And they also found polymetallic nodules may generate electro potentials that drive electrochemical reactions. And these results suggest a possible mechanism for DOP in the deep ocean. However, I think that we need more researchers to fully understand this process. Thank you. This is my presentations.